Welcome, curious minds, to another deep dive. Today we're embarking on a really fascinating journey into the very blueprint of life, our genetic material. Our mission really is to demystify genetic markers. What are they? How did scientists figure them out? And why have they been just absolutely crucial in understanding and maybe even manipulating life at its most fundamental level? We'll trace their evolution, starting with things you could actually see with your bare eyes, then move to biochemical clues, and finally we'll really dig into the DNA itself with a technique called RFLP.
And that journey is just so critical because these markers, they give us what some call a new, expanded, and augmented picture of genetic diversity. I mean, imagine trying to understand a really complex tapestry, but you don't know the different threads making it up. These markers are those threads. They're foundational for everything, improving crop yields, plant breeding, even conserving endangered species.
Yeah.
It all relies on understanding genetic diversity. They really are the signposts that have guided our understanding.
Okay, so let's establish our baseline then. When we talk about a genetic marker, what exactly is that? What makes something, a marker in genetics?
Right. Well, at its core, a genetic marker is just any detectable variation. Historically, these could be quite broad, from the visible traits of an organism, like flower color. Those are morphological or phenotypic markers. Then, as our tools got better, we moved to biochemical markers, looking at different proteins or enzymes. We even looked at the physical structure of chromosomes, that's cytogenetic markers. But for this deep dive, we're really going to focus on molecular markers. These are variations directly at the DNA level, things we can actually identify and, score. The key thing is they need to be polymorphic.
Polymorphic.
Yeah, meaning they show variation that lets us tell individuals apart. And ideally, they should be abundant and generally neutral, not under strong selection pressure. And of course, if they're truly genetic, they should follow those Mendelian patterns of inheritance, in the offspring.
That's a great distillation. So, okay, beyond just being detectable variation, why were they such game changers? For researchers, for breeders, what's the so what here?
Oh, the so what is huge. I mean, before these markers, we were pretty much limited to observing visible traits.
Yeah.
And those often don't tell the whole story, right? Genetic markers give us an entirely new and, frankly, far more detailed picture of genetic diversity, both within species and between them. This is actually how we build genetic maps. Think of them like biological roadmaps. They're essential for pinpointing the exact locations of genes responsible for traits. Simple ones, complex ones. They also enable really powerful techniques, things like associative mapping and genomic selection, which dramatically improves how we screen and select for desirable characteristics in, say, breeding programs. Ultimately, you can think of markers as the bridge.
A bridge.
Yeah, connecting the classical observations like Mendel's work with the cutting edge of modern molecular genetics and selection.
What a powerful analogy. A bridge between the old and the new. And I know we classify these markers in a few ways, right? One is how they're inherited. Could you walk us through the difference between, say, a dominant and a co-dominant marker? What does that really mean for what we can see?
Absolutely. Think of it like this. With a dominant marker, if an individual has even one copy of the dominant version, that's what you detect. You can't actually tell the difference between someone with two copies, the homozygous dominant, and someone with just one copy, the heterozygote. So if you're looking for a specific DNA band on a gel, maybe both show the band. You can't distinguish.
Okay.
But with a codominant marker, you can distinguish them. The heterozygote will show both versions. Maybe it shows two distinct bands, one from each parent. It's just much more informative for genetic analysis.
Right, more information.
Exactly. And the variations themselves, the polymorphisms we actually detect, they often come from tiny differences in the DNA sequence. It could be a single nucleotide polymorphism, a SNP, that's a change in just one DNA letter, or an insertion deletion, an indel, where a small piece of DNA is either added or taken away. These tiny changes can have pretty big consequences for how we actually read the genome.
So for our listeners, it really boils down to seeing those differences, right? Whether it's a visibly different seed color that's polymorphic, or if they're all the same color, which would be monomorphic, or maybe looking at patterns on a gel, seeing variation versus seeing everything look uniform.
Precisely. That variation is the key. It lets us tell one individual apart from another, which is, the whole point of using a marker in the first place.
Okay, with that foundation laid, let's take a little trip back in time to the very beginning, really, to the earliest and maybe the most straightforward type of genetic marker.
Right, these would be the morphological markers. Basically, the visual indicators of traits that vary between individuals. They're simply the characteristics we can see directly the outward signs of gene activity. Think about the physical attributes of a plant, the color, shape, size of its flowers or seeds or leaves, its overall growth habit, maybe even the presence or absence of pigmentation. And just like we discussed, depending on the specific trait, these can follow either dominant or codominant inheritance patterns.
It's actually incredible what just observing a pea plant could teach us. This takes us right back to Gregor Mendel, doesn't it? Around 1865, he basically invented genetics by meticulously tracking, what was it, 7 morphological traits in his garden peas?
Yeah.
Things like seed form, flower color.
Yeah, exactly.
He laid the groundwork for inheritance just by looking. It's amazing. And then later, I think around 1917, scientists saw the first case of genetic linkage in tomatoes. They connected a dwarfism gene to a fruit-shaped gene just by observation. And of course, Thomas Morgan's work, 1959, I think, with white eyes in fruit flies, that also relied on a morphological marker to uncover fundamental genetic principles. Mendel, bless his methodical heart, basically invented genetic mapping with just his eyes and, well, a keen sense of observation. Imagine the excitement when those patterns started to actually emerge.
Absolutely. It was truly revolutionary. And for a more practical, maybe relatable example, think about gossy pole glands in cottonseed. These are tiny little black spots you can actually see on the seed. The presence of these glands is a dominant trait, and this simple morphological marker can actually be used in what are essentially paternity tests for seeds.
Paternity tests for seeds.
Yeah, kind of. It helps breeders detect if a seed resulted from unwanted foreign pollen, or it can confirm if their controlled crosses were successful, basically detecting contamination. It's pretty low tech, but effective for certain things.
So while these morphological markers gave us our first real glimpse into inheritance, they weren't perfect, were they? What were the biggest challenges scientists faced when they were relying only on these visible traits?
You're right, they definitely had significant limitations. I mean, the major advantages were obvious. Simple identification, very low cost, you just need your eyes, basically. And they're typically functional, meaning the marker is usually part of a gene that directly affects the trait you see. However, they often aren't sufficiently polymorphic.
Meaning not enough variation.
Exactly. There just aren't enough clear, distinct variations to work with sometimes, especially if a trait is controlled by many genes, polygenic inheritance. And crucially, they can be heavily influenced by the environment or other interacting genes. That makes consistent scoring really difficult. And a big one is their distribution in the genome. They're not evenly spread out. They tend to be clustered in gene-rich regions, leaving vast stretches of the genome completely, well, unmapped.
Right. It makes you wonder how much was missed back then. That limitation must have pushed scientists to look for clues at a deeper level, beyond what was just visible to the eye. So our next stop on this journey moves into the realm of biochemistry, yeah?
Exactly. This brings us to isoenzymes. Sometimes people call them alloenzymes, too. Basically, these are homologous enzymes, meaning they do the same job in the cell, but they differ slightly in their structure. Amino acid sequence, usually. These subtle structural differences cause them to separate differently when you run them on an electrophoresis gel. That results in distinct band profiles, and those profiles are our valuable polymorphisms. The nice thing about isoenzymes is that they are generally codominant markers.
Ah, so you can tell the heterozygotes apart again.
Precisely. That gives you more genetic information. The variations themselves, they typically come from tiny changes in the gene's coding sequence, like point mutations or small insertion-deletion events that alter the protein structure or maybe its electrical charge.
Okay, so how did scientists actually use these biochemical differences? Can you walk us through the practical steps? How would they use isoenzymes in the lab?
Sure. It's a fairly straightforward lab procedure, relatively speaking. First, you take a tissue sample, usually leaves from a plant or good, and you make a crude protein extract. Just grind it up. This extract, which has all the plant's proteins mixed together, is then loaded onto an electrophoresis gel. Often a starch gel was used. The gel separates the proteins based mainly on their size and electrical charge. Once they're separated, the gel is incubated in a special solution. The solution contains all the necessary stuff reagents for the specific enzyme you're interested in to actually do its chemical reaction. And importantly, it includes a dye. The enzyme, if it's there and active, will catalyze a reaction involving that dye, causing it to change color and stain the gel, but only where that specific enzyme is located. So this lets us visualize the different allelic variants of the protein as distinct bands on the gel.
That's fascinating, like developing a photograph almost. So what were the main pros and cons of using isoenzymes compared to those earlier morphological markers?
Well, a major advantage, as we said, is their codominance. That's incredibly informative for genetic studies, much better than most morphological markers. They were also among the quicker and cheaper marker systems to develop back then. Required less specialized equipment than the DNA-based methods that came later. And actually, for some projects, maybe needing to identify only low levels of genetic variation, like quantifying mating systems in wild plant populations, they're still a viable choice today.
Still used.
Yeah,
Okay.
sometimes.
Yeah.
But they definitely come with their own set of limitations. There's a relatively small number of isoenzyme loci available in the genome that actually show useful polymorphism. You don't have thousands to choose from. Also, a bit like morphological markers, not all enzymes are active in all plant tissues or at all developmental stages, and their expression can sometimes be influenced by environmental conditions.
So that environmental influence is still a potential issue.
It can be, yes. And that means you might need to take several samples from a population over time or from different parts to get a really complete picture, which obviously adds significant labor.
Right, that makes perfect sense. So they provided a deeper look than morphology, but still had that potential environmental Achilles heel. But the real game changer, I guess, the moment we started truly deciphering the language of DNA itself, that came with our next marker type, didn't it? This is where we started using DNA as its own detective directly for the first time.
Absolutely. This really ushered in a whole new era with RFLP. That stands for Restriction Fragment Length Polymorphism. Here's the core idea. You take a DNA sample, right, and you use these special molecular scissors called restriction enzymes. Each type of enzyme recognizes and cuts DNA only at very specific short sequence sites. So the DNA gets chopped up into fragments of varying lengths. These fragments are then separated by size using gel electrophoresis, just like with proteins. Now, the key insight is this. If a mutation happens right at one of those restriction enzyme cutting sites.
So the enzyme can't cut there anymore.
Exactly. Or if an insertion or deletion event changes the length of a DNA fragment between two cutting sites, then the resulting fragments after cutting will be different sizes compared to the original sequence. For example, if a mutation causes an enzyme to miss a cut it would normally make, you'll end up with one single longer fragment instead of two shorter ones. These differences in fragment lengths, that's the length polymorphism part, those are our polymorphisms. And RFLPs are wonderfully codominant markers, again, offering really high informational content.
Okay, this sounds incredibly sophisticated compared to, looking at P colors. Can you walk us through the full process? It sounds complex. And what exactly is southern blotting? We hear that term a lot with RFLP.
It's definitely more involved, yeah. Step by step, it goes something like this. First, you carefully extract high-quality DNA from each sample. That's crucial. Then, that DNA is subjected to digestion by your chosen restriction enzyme, or sometimes a combination of enzymes. Let it cut. Next, you load those cut DNA fragments onto an agarose gel and run gel electrophoresis. This separates the fragments by size. Smaller pieces move faster and further down the gel. Okay, now here's where southern blotting comes in. You need to get that separated DNA out of the fragile gel and onto a more stable surface for the next steps, for probing. So first, the double-stranded DNA that's still in the gel is denatured chemically unzipped into single strands, usually using an alkaline solution. Then these single-stranded DNA fragments are transferred from the gel onto a special nylon membrane. It's like blotting ink. This transfer process is the famous southern blotting, named after its inventor, Edwin Southern.
Ah, okay. So that's the blotting part, transferring it to the membrane. What happens next? How do you see the differences?
Right. So once the DNA is on the membrane, it's permanently fixed there, immobilized. Then the membrane is bathed in a solution containing specially designed probes. These probes are short, single-stranded pieces of DNA themselves, and they're designed to be complementary to the specific DNA sequence you're actually trying to detect on the membrane. Traditionally, these probes were radioactively labeled, often with phosphorus-32, so you could detect where they stuck. They will hybridize, which just means bind, but only to their perfectly matching complementary sequences on the membrane, like a molecular Velcro.
Okay, so the probe finds its target.
Exactly. Finally, the membrane is placed against a piece of X-ray film in the dark. The radioactive signal from the hybridized probes exposes the film, creating an image called an autoradiogram. This autoradiogram shows distinct bands wherever the probe has bound. By comparing these banding patterns across your different samples side by side, you can identify the restriction fragment length polymorphisms, the RFLPs. It's worth noting, though, while radioactive probes were the original method, and obviously carried safety concerns, today we have much safer, alternative, non-radioactive stains. Things like digoxygenin or fluorescein can be used instead.
That's a huge methodological leap for sure. But the idea of these probes is still a bit mysterious. How do scientists even develop these specific DNA sequences to probe for? It seems like a bit of a chicken and egg situation. You need to know what you're looking for to find it, right?
That's a great question. And yeah, it was definitely a challenge, especially early on. One common method involved digesting the total DNA from the organism with specific enzymes that are sensitive to DNA methylation. See, genes, which are often the expressed sequences researchers are interested in, tend to be unmethylated in many organisms. So this digestion strategy would effectively enrich the sample for those gene-rich regions, kind of filtering out some other stuff. Then, DNA fragments of specific sizes, maybe between 500 and 2,000 base pairs long, would be carefully cut out of a gel, extracted, purified, and then cloned into a plasmid vector, basically making many copies of that specific fragment.
Creating a library of potential probes.
Exactly. These cloned inserts, these potential probes, would then be individually screened against the organism's total DNA. The goal was to select only those clones that hybridized to single or low-copy sequences in the genome. You want your probe to be unique enough, or nearly unique, to give a clear, specific signal on your southern blot, not light up everywhere. Also, sometimes researchers could use heterologous probes.
Heterologous, meaning from a different species.
Yeah, exactly.
Yeah.
Probes developed from a closely related species could sometimes work if the DNA sequences were similar enough, conserved enough through evolution. That could save some development time.
Okay, so after all that intricate lab work, what was the final verdict? The report card on RFLP, what were its major strengths and, I guess, its weaknesses?
RFLP definitely offered significant advantages at the time. They're highly informative because, as we said, they're code-dominant, and they can potentially reveal multiple alleles at a single locus. That tells you a lot about genetic variation. They're often associated with actual gene regions, making them potentially functionally relevant, not just random markers. And it's known as a very robust methodology. When done right, it gives good reproducible results. However, the disadvantages were also pretty substantial. Okay, well, RFLP is notoriously low throughput. You typically detect only one or maybe a few loci per assay, per experiment. That makes large-scale mapping really slow going. You either needed to already know some sequence information to design your probe, or you had to invest heavily in developing a suitable probe library, like we just discussed. That takes time and effort. The entire process was incredibly laborious and expensive. Just think of all the steps: probe labeling, DNA cutting, running gels, blotting, hybridization, washing, waiting for autoradiography. It's a lot. As we mentioned, the original use of radioactive probes made it relatively dangerous work. And importantly, in the pre-PCR era, before we could easily amplify DNA, it required a fairly large amount of high-quality starting DNA, which could be a real limiting factor depending on your samples.
Wow, yeah. Laborious, expensive, slow, potentially dangerous. You can see why scientists were keen to find alternatives eventually. We've truly journeyed through time and technology today, haven't we? From just looking at the color of a pea to, dissecting DNA fragments with enzymes and radioactive probes. It's really an incredible evolution of techniques. So we've seen how markers evolved from these simple observations to quite complex molecular techniques. But what does this all mean for today's research? Do researchers typically integrate all these different marker types, morphological, biochemical, and the various molecular ones like RFLP or newer ones, into a single big analysis?
That's an excellent question, and it's something people often wonder about.
Theoretically, yes.
Integrating all marker types could significantly increase the resolution, the detail, and the saturation of genetic maps. It could give an even more complete picture. However, in practice, it's actually quite rare.
Oh, why is that?
Well, there are several key reasons, really. Firstly, the expertise required for each type of marker is often highly specialized. Labs tend to focus. They might be experts in DNA markers or maybe protein work. It's unusual for one lab to maintain all the diverse skills, equipment, and reagents needed for morphological scoring, protein analysis, and DNA work all at once. Just very specialized stuff. Secondly, cost is always a factor, isn't it? DNA markers, especially newer high-throughput ones, while powerful, can still be expensive to run on many samples. For many specific research questions, maybe simpler morphological or isoenzymatic markers actually provide sufficient information and at a much lower cost. So why overspend? But maybe the most critical reason, and it highlights a huge advantage of DNA-based molecular markers, is that they generally show no interaction with the environment.
Unlike the others we discussed.
Exactly. The DNA sequence itself is constant. It doesn't usually change based on temperature or what tissue you sample or the plant's age, unlike, say, phenotypic traits or enzyme expression levels, which can vary quite dramatically. This makes DNA markers incredibly reliable and consistent across different conditions and experiments. That's a massive advantage.
That's a really crucial point, reliability. So, as you, our listener, consider all these different ways we can mark genetic information, think about how that choice of marker, Whether it's something as simple as the visible difference in a pea's color or the intricate pattern from a restriction fragment length polymorphism, think how that choice can fundamentally shape what we discover about an organism. And, maybe more importantly, how we then choose to improve it, whether it's a crop or understanding a disease. What stands out to you as the most surprising or maybe the most impactful insight from our deep dive today?